A group of medical practitioners visited a school as a part of celebrating International Kidney Day to create awareness about the importance of kidneys to our overall health. We know that the blood is filtered through the kidneys and excreted as urine. But are you familiar with the internal mechanism of urine formation? In this video, we shall discuss the mechanism of urine formation. Recall that the nephron is a basic unit of the excretory system. Nephrons are the functional site for the purification of the blood. For the purification process, there are generally three major steps involved in urine formation: glomerular filtration or ultrafiltration, selective reabsorption, and tubular secretion are the major processes involved. So let us study each one in detail. Before understanding the mechanism, we should be thorough with the structure of a nephron. A nephron has a renal corpuscles, that is a glomerulus within the Bowman's capsule, a proximal tubule which has convoluted and straight components, an intermediate tubule, that is a loop of Henle, and a distal convoluted tubule along with a connecting tubule and cortical outer and inner medullary collecting ducts. In glomerular filtration, the blood undergoes filtration through the pores of the glomerulus in the Bowman's capsule. You need to note that the size of the efferent vessel is less than that of the afferent vessel. Because of this difference in the artery diameter, blood leaving the glomerulus creates a hydrostatic pressure. Let us now learn about the different layers: the endothelium of the glomerular blood vessels, the epithelium of the Bowman's capsule known as the podocytes, and the basement membrane between these two layers. The force will be higher inside the glomerulus, and this pressure forces the blood to leave the glomerulus, resulting in filtration of blood. For effective functioning, the layer of podocytes has spaces called filtration slits or the slit pores. The filtrate contain essential and non-essential materials. This filtrate is then collected in the capsular space of the Bowman's capsule, and this filtrate is termed as a glomerular filtrate. The term ultrafiltration refers to the fine filtering of blood through three-layered membranes. Is there any way to quantify the rate of filtrate? Yes. The amount of filtrate produced by the kidneys every minute is known as the glomerular filtration rate or the GFR. The glomerular filtration rate of a healthy individual is approximately 125 milliliter per minute, that is 180 liters per day by two kidneys. Have you seen cellular modifications in the point of contact between the distal convoluted tubule and the afferent arteriole? They are the juxtaglomerular apparatus or the JGA. These are the signs of the regulation of the glomerular filtration rate. What happens when there is a fall in this rate? When the rate falls, it activates the juxtaglomerular cells to release renin, which further stimulates the glomerular blood flow and thereby makes the glomerular filtration rate back to normal. Now, we can infer that the blood is filtered. But are you familiar with the rate of the reabsorbed content? The renal tubules reabsorb 99% of the filtrate. Useful components such as water, electrolytes, and organic nutrients like glucose, amino acids, vitamins, hormones, etc. are selectively reabsorbed from the filtrate back into the blood in the proximal convoluted tubule as it travels to the renal tubules. Positively charged ions are also reabsorbed by the active transport, while the negatively charged ions are reabsorbed most often by the passive transport. Large amounts of the active reabsorption of nutrients, water and the electrolytes occurs in the proximal convoluted tubule or the PCT, and the nitrogenous waste are absorbed by the passive transport. A low rate of reabsorption takes place in Henle's loop or the loop of Henle. Water is mainly reabsorbed from the collecting tubule or the collecting duct through the process of osmosis. Along with the process of reabsorption, the secretion of substances also takes place in the nephron. The filtrate then finally acquires the secretion of excess hydrogen ions, ammonia and potassium ions from the proximal convoluted tubule, that is a PCT, and the distal convoluted tubule, that is a DCT. Tubular secretion of hydrogen ions, that is the H+ ions, is very important in maintaining the normal blood pH. Recall that Henle's loop is involved in the transport of water. The tubular filtrate is finally known as a urine. Human urine is usually hypertonic. The urine is then transported from the collecting duct to the ureter through the pelvis. The urine is stored temporarily in the urinary bladder. When the urinary bladder is full or filled with more than 300 milliliter of urine, a pressure is exerted on the wall. This generates a sensation to urinate and the urine is removed from the body through the urethra. Thus, the micturition process occurs. Let me ask you a question. Why does urine have an yellow color? The color is due to the presence of urochrome pigment. It is a breakdown product of hemoglobin. Now, it's time to check the major components of urine. The major part of urine is water, which is 95%, urea, which is of 2%, uric acid, creatine, pigments and inorganic salts, which comprises 3%. How does the PCT also helps maintaining the body fluid pH and ionic balance? Which part of the nephron maintains the high osmolarity of medullary interstitial fluid? Which part of the nephron balances the sodium-potassium balance in the blood? We shall discuss all these questions in the next video. Now, let us summarize what we have learnt.
Manorama Horizon. Learning made simple.